Emily graduated with a bachelor's in biology from Penn State Behrend, and then just recently, this December, completed her master's work in horticulture, where she worked on a fungal pathogen that was originally isolated from the Penn State Grape Research Center out in Northeast PA. It's a very interesting story. So without ado, I'd like to introduce Emily's talk on implications of the emerging pathogen Gnomoniopsis castaneae for domestic Fagaceae species.
Thank you for that. So yeah, I'm going to be talking a little bit about the results of some of my thesis research to investigate this emerging pathogen and what it might mean for domestic Fagaceae species such as oak, beech, and chestnut. I'm going to make you smaller here. All right. Gnomoniopsis castaneae, also known as Gnomoniopsis smithogolvii, is a fungal endophyte, saprophyte, and pathogen most commonly associated with chestnut. Right now, as a pathogen, it's considered to be host genus specific to chestnut species. And its most readily recognizable symptom in its pathogenic form is as brown rot of chestnut. So it rots the soft tissue of the nut. And this is why it's more commonly known as chestnut brown rot, which is certainly a lot easier to say than Gnomoniopsis castaneae. Now, cases of brown rot in chestnut go back as far in time as the chestnut has existed. There has always been some kind of agent causing brown rot of the chestnut tissue. But in the 1980s, we started to see an increase in incidence, and this really took off in the early 2000s and really began to negatively impact the crop yield of chestnuts year-round. And so in the early 2000s, researchers out of Europe and Australia really wanted to try and figure out what could possibly be causing this, what was the causal agent. And so the pathogen, or a pathogen, was simultaneously described in Italy and Australia in the summer of 2012. Just a few weeks apart, these publications were put out. So the researchers out of Australia named it Gnomoniopsis smithogolvii, while the researchers out of Italy named it Gnomoniopsis castaneae. Both of these researchers, both of these projects identified this pathogen as a really concerning agent of nut rot, or even the primary agent of nut rot in Castanea species. The problem came when they realized that both of these described the same pathogen. And so because they were published so closely together, there's a lot of debate over who has the naming rights and which is the valid name of the pathogen. But because Gnomoniopsis castaneae is pretty widely accepted throughout Europe, and that's where the majority of the research has been, that's what I'm going to be calling it moving forward in this presentation. So as I said, it's a pretty big issue in terms of nut rot, and it's caused a lot of economic losses throughout Europe and Oceania. But it's also been identified as a canker agent of chestnut. It was initially identified in 2015 by some researchers in India who thought that what they were looking at was a case of chestnut blight infection, which is another notorious canker-causing agent for chestnut species. However, they found that it was this pathogen instead. Subsequent research out of Switzerland and out of the UK supported their findings. But right now, this pathogen is still really considered to be host genus specific and really thought to only be significantly damaging to chestnut species. However, some research out of Europe has found it through casual research that it was associated, it acted as an endophyte or as a mild pathogen of multiple other hardwood species. And that includes oak, manna ash, hazelnut, and common box. And importantly, it was mildly pathogenic in the hazelnut and common box, and it acted as a canker agent in those hardwood species. So this kind of brings into question whether this really is a host-specific pathogen. Since its discovery in 2012, this really almost seems to be a pathogen on the move. It has popped up all across Europe, Asia, and then in 2019, it was described in the United States for the first time as an agent of nut rot in some Michigan chestnut orchards. It's also been documented in Chile, although there has been no official first report, which is why you don't see it on this particular map, but it has been found in both North and South America. So you might be looking at this map and thinking, well, that really looks like it's more of a problem for Europe, maybe a problem for Australia, New Zealand, but not really something that we should be concerned about here. Probably don't even know that much about chestnuts. Well, up until the turn of the 20th century, the American chestnut really was an important tree for our country. It was an American icon. Prior to the turn of the 20th century, it accounted for approximately 25% of the canopy along its native range in the Appalachian Mountains. So that's 1 in 4 trees was thought to be an American chestnut. These trees were really important because they had this prolific annual nut production, this annual nut crop that was vital to not only forest animals, but to people who lived in the Appalachian Mountain range, because they could use this food for themselves and as fodder for their cattle. And this was really reliable. Every single year, the chestnuts would put out these huge crops. Not only did they have this wonderful nut crop, but they also had rot-resistant wood, which was very valuable. You can still find it in some places as fencing. Tables are really sought after. It's stuck around, it doesn't rot. And it also supported a large tanning industry. But that was until the introduction of chestnut blight. So chestnut blight was introduced to the United States just before the turn of the 20th century, and it was introduced from Asia. So the American chestnut at that time, and still doesn't, have any known natural resistance, or very little natural resistance to the pathogen. And what this means for the American chestnut was that it was very easily infected and very susceptible to damage. And within a period of about 50 years, an estimated 4 billion trees were wiped from the landscape. So let me get my little laser pointer here. You can see in the top of the screen here is the estimated range of what the American chestnut was prior to the introduction of chestnut blight. And to give you an idea of just how devastating this pathogen is, this is an image taken in 1930 of the Chattahoochee National Forest in Georgia. And this is a strictly chestnut forest. So you really get an idea of how devastating this would have been to our forest systems. Almost immediately, there was a lot of work by researchers and by passionate citizens to try and prevent the American chestnut from slipping entirely into extinction. And now it's considered to be functionally extinct. It exists in our forest systems, though it rarely grows large enough to reproduce. So it's out there, it's just not reproducing. So these efforts have been ongoing since the very beginning, but it really took off in the 1980s when there were a group of scientists and citizen scientists who worked towards introducing a breeding resistance program where they crossed the American chestnut with the more blight-resistant Chinese chestnut. So they hoped that through a series of crosses and back crosses, they could produce a hybrid that had that stature and that nut production of the American chestnut, but with a level of resistance that was similar to what the Chinese have. There were also efforts to introduce hypovirulence into the pathogens population. So that would be introducing a virus that effectively prevents it from causing damage. Likewise, there have been efforts with creating transgenic chestnuts in which they've had the oxidase gene stuck into their genome, and that effectively shuts down the chestnut blight's oxalic acid production. So it diminishes its ability to cause the damaging infection that it had previously. But all of these efforts, all of these groups had the one big goal of restoring the American chestnut to its native range, but that has not yet happened. This is still an ongoing effort. And so right now, in the 100 years or so since the American chestnut has really been lost in our forest systems, other important nut producing trees such as oak or beech have taken the place that chestnut once held. One of these groups, the American Chestnut Foundation, which really focused heavily on the breeding program, provided the Lake Erie Regional Grape Research and Extension Center in Northeast with 15 chestnut seedlings in 2013. So they did this as part of their ongoing outreach and monitoring program where they wanted the seedlings to be planted and then to monitor the health and success, particularly of the hybrids. And we had the American chestnut and the Chinese chestnut for comparison. In the summer of 2018, we were tasked with observing these trees and just kind of describing the health and seeing how they were doing, how they were progressing. And we noticed that several of the American chestnuts were battling some kind of illness or attack. They were sending up shoots from their bases. They had yellowing, wilting leaves, and 2 of them had very large cankers of the trunk. Now, initially, we assumed that most likely what we were looking at was a case of chestnut blight infection, because that is the most common infection for chestnut trees to cause cankers, particularly the American chestnut, which is where we saw the most pronounced symptoms. But as you can see here on the left-hand side of the screen, this is a pretty standard looking chestnut blight canker. And these tend to be rust colored or a little orange, and they're sunken. These will girdle the tree, meaning they'll cut off transport between the roots and the shoots. So there's no more nutritional transport. What we were seeing on our trees was a little different. Some of them were girdled and sunken. Some of them had blown outwards. They were a different color. So we weren't entirely sure what we were looking at, but we assumed that we were probably looking at simply an atypical presentation of a chestnut blight infection. But we wanted to confirm our suspicions. So we took samples from the margins of the cankers and we isolated pure fungal cultures from those. And then we sequenced the ITS region of the DNA. And blast identification showed us that we had not in fact isolated chestnut blight from our cankers, but instead we had isolated Gnomoniopsis castaneae, or Gnomoniopsis smith-ogilvii was the closest identified blast. And so this certainly raised a lot of questions for us in terms of what we were looking at. And in the years that followed, we made some trips down to University Park to their chestnut test orchards down there, and we found that some of their chestnuts also had Gnomoniopsis castaneae infections, and that is the isolate that you see at the center of the screen here. And then we were put into contact with a gentleman out of the University of Maryland who had also isolated the pathogen from some of his chestnut trees in 2016, 2017. So what this was telling us was that in the mid to late 2010s, there was all of a sudden a lot of discovery of this pathogen in the United States, and we really didn't know what that meant. Was this introduced? What would that mean to our chestnut trees, and what could it mean for other close relatives of the chestnut? So our primary research objectives for this project were to evaluate the cankering and nut rot potential of these 3 domestic Gnomoniopsis castaneae isolates in multiple Fagaceae species. So we wanted to look at multiple Fagaceae species because we saw out of Europe that there is some evidence to suggest that other hardwood species might be susceptible to a damaging infection. And we wanted to know specifically if oak and beech might be susceptible. So we completed these, approached these research objectives through a series of nut, branch, and stem infection assays. We began with our nut infection assays in the fall of 2020 by collecting oak acorns from multiple locations around Erie County. So we collected red, pin, and white oak acorns, and we were provided with chestnut seeds from central Pennsylvania chestnut orchards. We had American, Chinese, and hybrid chestnuts. Unfortunately, there was a pretty dramatic drought in the summer of 2020, so the chestnut crop was affected, and we weren't able to utilize as many nuts for our study as we would have liked. But we did want to include them because we know that the primary symptom of this pathogen is chestnut brown rot. So we began with surface sterilizing all of the nuts to remove any potential external contamination. And then we used a bone marrow biopsy needle, which is the tool that you see right here, to create a 1 millimeter wound through the shell and into the soft tissue of the nut. And into that wound, we put either a 1 millimeter plug of one of those 3 live fungal strains or a control sterile agar. And then we wrapped each nut individually and entirely in parafilm to prevent cross-contamination and desiccation. And then we stored them in these little plastic containers here. So each of these containers would hold representatives of one genus. So they would have one representative of one particular genus. In this case, these are all oak. And it would be one treatment. So we had 25 replicates of each species per treatment within the oak. We only had 11 replicates of each of the chestnut species, and that was due to that significant drought and the lack of availability of more nuts for a greater number of replicates. This test was repeated once. The nuts were stored for a period of 1 month at 25 C. For our branch infection assays, we wanted to more fully describe the response of the tree tissue to the presence of the pathogen on a year-round basis, so during multiple seasonal tissue stages. So we began by collecting branches in November of 2020 when the trees were in their endodormant stage. So this is when they're deeply dormant. This is prior to the completion of chilling hour requirements. Then we collected again in February of 2021 when they had completed those chilling hour requirements, and now they're eco-dormant. So they're a little more environmentally responsive. They're just waiting for the right cues to entirely exit dormancy. And then finally, we collected in March of 2021 when the trees had exited dormancy and had resumed active growth. They were beginning to leaf out. For our branches, much as with our nut infection assays, we again surface sterilized, and then we cut each branch to 15 centimeters in length, and we wrap them entirely with parafilm for the same reasons as with the nuts. And then we used a cork borer to create a 5 millimeter wound at the midpoint of each branch. And that wound was filled with either one of those 3 live fungal treatments, the live fungal strains, or a heat-killed control representative of one of those strains. So we had autoclaved representatives of each of the isolates to kill the fungus and get an idea of whether the tree tissue was simply responding to the presence of the proteins of the pathogen or whether it was responding to an actively live damaging pathogen. So here we again had the same species that we utilized for our nut infection assay. So the red, pin, and white oak, the American, Chinese, and hybrid chestnut, but we also included beech. Although we were not able to include them in our nut infection assays because there wasn't a crop for it that year, we did want to be able to more fully describe what might be occurring within the Fagaceae family. So we included them for our branch assays. Each branch was packaged individually into a zip top bag, and we stored those at 25°C for a period of 1 month. This was completed only once for each of those tissue stages. And we had 6 replicates of each of the treatments per species for both of the dormancy stages and 8 replicates for the active growth. But we also wanted to look at the response of a live, actively growing seedling to the presence of this pathogen. We didn't want to just be looking at tissue that had been cut from a tree and kind of use that to gauge how the tree might respond. We wanted to see what's going to happen if it's still alive and can potentially fight off this pathogen. And so we set up seedling infection assays. And we began by planting seeds of all of the chestnut species and red oak in January of 2020. These were later culled in June of 2021. We didn't include any of the other oak species because at the time that we had planted out this seedling plot, we hadn't intended to include any more. So, but at least we had the one red oak. For this set, we again followed the same kind of methods that we use for our stem infection assays, where we used a cork board to create a 3 millimeter wound at about 10 centimeters above the soil. And we only utilized the Erie live fungal culture and the Erie heat-killed control for this test because we were attempting to complete Koch's postulates, demonstrating the pathogen as a causal agent of the cankers that we had observed on our trees initially in 2018. So these were wrapped up and they were allowed to continue to grow in ambient greenhouse conditions for a period of 2 months before they were cut down and evaluated. So evaluation of the induced rot depended on the tissue that we were looking at. For nut rot, we couldn't be terribly precise because nuts vary pretty greatly in size. And so what we did was we looked at our nuts and estimated the area of rot on a scale of 0 to 4, with 0 indicating that no rot is present and 4 indicating total rot of the soft tissue, which you can see an example here of kind of what our sliding scale would look like. For our canker measurements or the lesions, we were able to be a little more precise. For the branch and stems, we stripped the bark entirely from them, and then we measured the longest point and the widest point of that lesion and then calculated the area of it as an ellipse. For our stem infection assay, we also wanted to know whether this pathogen had the potential to cause damaging girdling, much as chestnut blight is known to do. And so we did this by, again, using a ranking scale of 0 to 4, as we did with the nut infection assays. So a 0 would indicate that there was no girdling at the point of infection. It was entirely smooth there, whereas 4 would indicate that the girdling had gone all the way around the seedling stem and was cutting off nutrient transport between the root and the shoot. So for our data analysis, we wanted to really We wanted to look first at whether treatment or species were significant predictors of rot, but we also wanted to evaluate whether the individual fungal strains were significant predictors of rot. And we ultimately found that the individual fungal strains were not, but treatment and species were. So what you will see moving forward in their results is that all of the fungal treatments have been pooled together, simply labeled as Gnomoniopsis castaneae. Likewise, with the controls in the branches, you will see that they have been pooled. So for our nut infection assays, what you are looking at here is a frequency plot. So this gives you an idea of how frequently a particular ranking occurred for a treatment and species. So we have frequency on the y-axis and treatment on the x-axis with the chestnut and oak species. For this, we utilize post hoc Tukey tests with Bonferroni correction at a reduced alpha for the interaction. And what we saw here was that there was significant rot induced in the American chestnut and all three of our oak species. And this was a little bit surprising because there hadn't been any such rot noted in any previous studies with oaks. For our branch infection assays, treatments were pooled by species. Because the data was non-normally distributed, we did have to utilize a Box-Cox transformation of the canker area, and I put our transformation values at the top because it did vary depending on the seasonal tissue state. But all of the figures that you're looking at are back transformed for easier interpretation. We utilized the two-way ANOVA with a Bonferroni correction for that interaction between treatment and species. Here on our y-axis, we have the canker area in millimeters squared. On the x-axis, we have our tree species, and then our treatments are orange for the control and a green for the live fungal treatment. And what we found with our endodormant was that the red oak and the American chestnut were susceptible to damaging lesions, damaging infection prior to the completion of those chilling hour requirements. So this was tissue that was collected in November of 2020. This would be if conditions are favorable. We need to remember that these were put into an incubator. They weren't at ambient outdoor temperatures. For our ecodormants, we saw again that red oak and American chestnut were both susceptible to damaging infection, but now we saw the addition of Chinese and hybrid chestnuts. So this is following the completion of those chilling hour requirements. These are tissues that were harvested in February of 2021. And finally, with the tissue harvested in March of 2021, when the trees had resumed active growth and were starting to leaf out, we saw that red and pin oak, as well as all three chestnut species, experienced significant lesions as a result of the live pathogen compared to the controls. So what we saw really through this test was that susceptibility varied by season and by species, so the seasonal tissue stage. For our stem infection assays, again, we did have to utilize a Box-Cox transformation. You are looking at the back transformed data. We have canker area in millimeters squared on the Y and species on the X-axis with the same color coding for the treatments. We again utilize the two-way ANOVA with a Bonferroni correction for the interaction. And here, interestingly, we found that all of the evaluated tree species, so that's all three of our chestnut species, with the addition of the red oak, had significant cankers induced by the live pathogen compared to the control. So just to give you an idea of what this looked like when we were tearing down that test, what you have here is on the top, these are the seedling stems in situ, and on the bottom, this is following the removal of bark. We have the control on the left and then the live fungal treatment on the right for each of these, with the American, Chinese, hybrid, and then red oak across the top. So you can kind of get an idea of just the kind of damage that this pathogen was causing, the live pathogen was causing to these seedlings and what that might look like. For our stem girdling, this is the last little piece of our data analysis here. We again are utilizing a frequency plot to give you an idea of how often we saw different rankings of 0 to 4, with 4 being the most significant girdling and 0 being no girdling present. Here we utilized that post hoc Tukey test with a Bonferroni correction, and we saw that there was significant girdling induced in all of our tree species, including the oak, with the live treatment compared to the control. So what this really tells us is that, at least under lab conditions, Gnomoniopsis castaneae induces significant rot in multiple Fagaceae species, not just in chestnut. We saw that there was significant nut rot in all 3 of our oak species and the American chestnut. We saw that there were consistent significant lesions on the red oak and the American chestnut branches, and that significant lesions on all of our seedling species. So the live seedlings all saw significant damage. We also saw that the canker responses differed between the seasonal tissue stages, and this could be really important as we continue to experience warming winters, which might contribute to a greater incidence of canker damage. If we have warm spikes in the middle of the winter where the pathogen has the opportunity to grow, but the trees aren't active enough to combat that infection, we may see more canker damage. But importantly, we did see that none of these strains differed in their nut rot or cankering potential. So we're not seeing that one of these strains is more virulent than another. And this altogether has a lot of implications for our forest systems and for the American chestnut. This certainly brings into question the host specificity of the pathogen, which had previously been considered to be host genus specific to chestnut, but now we're seeing that, at least under lab conditions, it can be very damaging, significantly damaging to multiple oak species. And this could be a significant ecological risk for our forest systems, which are still recovering from the losses of the American chestnut and are already dealing with regeneration issues from oak. So this pathogen has the potential to infect oak and cause damaging nut rot or damaging cankers out in our forest systems. It's really something we should keep our eyes on. But it really has a lot of implications for the American Chestnut Restoration Project too. That's something that citizens, scientists, the government have sunk more than 50 years of efforts into. They've sunk millions of dollars into not only preventing its extinction, but trying to restore it to our forest systems. But with the presence of this nut rot pathogen and the kind of damage that we see that it can cause to the American chestnut, it raises a lot of questions about the potential for the future of that program and where we go from here. And with that, I would like to thank my advisor, Dr. Michael Campbell, Dr. Ivor Knight for providing a lot of the financial support for this research, Sarah Fitzsimmons and Bruce Levine for providing chestnuts and samples for us to utilize in this study. And I'm open to questions.
Thank you, Emily. Great seminar. Are there any questions for Emily?
Emily, this is John Carlson. Have there been any reports of infections outside of the Great Lakes or outside of the Erie area yet for this?
Yes, so we know that it's been identified in Michigan, 2 locations in Pennsylvania, Maryland, and there at one of the Chestnut Foundation meetings a few years back, there was talk of it being identified in Virginia. So we know that it's at least in 4 different states in the United States right now.
Okay, gotcha. Oh, sorry.
Hello, Emily. This is Raymond. Is there any information on like what might be the vectors? Is this a soil-borne pathogen? Is this carried by human activity or anything else?
Right now, it's looking like wind dispersal is a major factor in how this is spread. There has been a lot of debate over whether the Asian chestnut gall wasp plays a role, but it doesn't look like that's able to carry viable inoculum. So right now, it's most likely wind dispersal that's carrying this.
Thank you.
Me again, sorry, you haven't tested the transgenic chestnut?
No, we have not, no, and that's something we hope to do. We don't know right now whether Gnomoniopsis castaneae utilizes oxalic acid to cause the damage, or if it's something else. Most likely it is oxalic acid, which would be good for the transgenics because that's really what they negate.
Yeah, no sources of resistance yet identified naturally though, so.
Yeah, no, yeah, chestnuts are having a lot of struggles, unfortunately, but I think transgenics are probably a really good future for them, and I think I would suspect that this pathogen utilizes oxalic acid, and the transgenics will be able to combat it.
Good.
All right, we got a question in the chat. It says, would you say the severity of this pathogen could be as bad as chestnut blight, or is it less of an issue? Well, that's hard to say. I think what we've seen so far is that it's not nearly as bad as chestnut blight, but it's not terribly studied outside of Europe. So I think it certainly warrants more research in the United States to see what it could mean here. We didn't do a side-by-side comparison, so I really can't say what chestnut blight looks like directly compared to Gnomoniopsis castaneae, if that answers your question.
Okay, I guess if there's no more questions, I'd like to say thank you very much for Emily's talk. It was nice work. Yeah, I do believe we have some plans to do a test on the transgenics coming up. So that's something that's going to get done. So thanks, Emily.
Thank you.